RNA immunoprecipitation in tandem followed by sequencing, or RIPiT-Seq, is a method for identifying what regions of RNA are bound by a particular pair of RNA-binding proteins. The most unique advantage of RIPiT is its ability to target two different proteins in two purifications. This provides a powerful way to enrich for a compositionally distinct RNA-protein complex from other complexes. Also, RIPiT-Seq is UV cross-linking independent and can be applied to many proteins that act on RNA without directly binding it.
While this method does not directly extend towards therapy or diagnosis, RNA binding proteins and RNA processing have been associated with several diseases like neuropathies and cancer. RIPiT-Seq provides a tool for studying functions of disease-linked RNA binding proteins. RIPiT-Seq requires a system where an epitope-tagged protein can be expressed. It has only been tested in HEK293 cells and is most amenable to cell culture. However, now, with our ability to tag proteins using CRISPR, RIPiT-Seq can be applicable to diverse model systems. The success of RIPiT depends upon two efficient immunoprecipitations. So it will be critical to optimize immunoprecipitation conditions for the antibody being used. Also, working with RNA requires proper precautions, so RNase-free reagents should be used during and after RNA extraction.
Demonstrating the procedure will be Lauren Woodward, a graduate student in my lab.
Start by washing monolayer cells gently with 15 milliliters of chilled PBS per 15 centimeter plate. Then, scrape off cells into 30 milliliters of PBS and collect them in a 50 milliliter conical tube.